to this special Cancer Center seminar. Delighted to welcome our guest, Dr. Jacob Scott from Cleveland. I met Jake about a year ago where he was an invited speaker in Abu Dhabi for a wind consortium.
We actually met on the plane.
Oh, that's right.
In Newark on the way.
That's right. And we do have its document.
Yeah, that's right.
It's out there.
Oh, I should have pulled that tweet. Yeah. You can pull it up during this one.
Yeah. So Jake is a physician scientist and professor of radiation oncology at the Cleveland Clinic, an associate director for data science at the Case Comprehensive Cancer Center. He specializes clinically in the treatment of patients with sarcoma, and his lab studies the evolution of therapy resistance in cancer and pathogens. So he's really an expert on tumor evolution and resistance to therapy. But there are many really interesting and unique things about Jake that I'll tell you about. So he works in the space of mathematical radiation biology, where he's come up with these concepts and algorithms, and he'll tell us how they're being applied in the clinic. Genomic adjusted radiation dose, something called GARD, and temporally feathered radiation therapy, TFRT, which is work really in the field of precision radiation oncology. So another thing, even though we met a year ago, I learned yesterday that Jake is a veteran of the U.S. Navy submarine force before he became a physician scientist. And I think, maybe he'll say something about that, how that influenced him. And so he did his medical school at Case. He did a residency in radiation oncology at the Moffitt Cancer Center. And then he says he felt frustrated with the one-size-fits-all approach in modern oncology. So he took a hiatus to get a PhD at Oxford in mathematical biology. And his thesis focused on the role of heterogeneity in evolution, both genetic and microenvironmental, on cancer evolution and radiation response. Board certified. He's very well funded and publishes in high impact journals. He currently has an NCI U54, R37, and U01. So today he's speaking about towards personalized medicine for all, and how he's using molecular tools of AI for radiation and chemo adjustments. But before we start, Jake was hosted yesterday by CCMB and by Jake Margul, an MD PhD student who's going to give you a little momentum.
Oh, I was getting nervous.
Wait. Come over, we got to do like the photo op over here. Okay. Good, you've got your brown shirt. Excellent.
Where's the who's taking a photo? You?
I guess. Well, I can do a selfie of us.
Yeah, there you go. There you go. To the brown mug. Hold on, wait.
It's the Legorreta Cancer Center, I believe. Hold on.
Oh yeah, it's okay. Thanks for inviting. Both.
Look forward to your talk.
Cheese. Oh, I'll put it right here. Thanks. Thanks for that really kind introduction, Wafik. I think I'm really humbled to be invited to Brown. It's my first visit to Providence, and yesterday, this whole visit's been a real blast for me. I got yesterday to hang out in CCMB and EEOB, Evolution Ecology and Organismal Biology, and meet some people whose work I've been reading for 2 decades. And I think the same is true here. Wafik's work and many others. I feel incredibly privileged today to be an academic, seeing the world and having the conversations that we have over decades together in our written communications and our thought it's really an amazing career to be able to think deeply about the things that plague humanity or the fundamental natural phenomena that are all around us and so beautiful, and to just talk to really smart people about it. And I'm hoping that I can maybe open your eyes to some of the problems in our field, in oncology and radiation oncology in particular, and some chemotherapy ideas and how evolution shapes our thinking. So I'm going to give a talk in 3 parts today. The beginning is going to be a little bit of my own scientific philosophy and what our group does and thinks about broadly. If you're at my talk yesterday, it's unchanged from the beginning of that talk, so apologies, or close to unchanged. It's about 10, 15 minutes. And then I'm going to dive into a little bit of nitty gritty rad onc and rad bio mathematics. And I'm going to show you how I think we've opened the door for personalized medicine and radiation oncology. And that's really important because half of all cancer cures are based on radiation. And if you could improve radiation by 5%, it would be more cures than targeted and immunotherapy put together. So think about that. It's also just worth mentioning that 5% of the NCI budget is radiation research, and half of cures come from there. And it's an interesting imbalance, I think. But I think it's partially our fault as rad oncs because we're happy in the basement. We're happy doing what we do. And we are quite comfortable. And I'm going to, during the course of this talk, I hope show data that makes you uncomfortable with what we do, because you can't change if you're comfortable. You have to get uncomfortable before change occurs. And so I'm going to try during this talk to make people a little bit uncomfortable with the state of oncology. That said, I think we do a great job, but we have room to improve. My title is a huge mouthful, Towards Personalized Medicine for All, Building Phenotype-Focused Transcriptomic Models of Therapeutic Response to Provide Decision Support Tools for Radiation and Cytotoxic Chemotherapy. I tried to get as many buzzwords in as possible to fill the seats. So if you're a transcriptomic person, don't worry, I got you covered. If you're a precision oncology person, I got you covered. If you like evolution, that's the way I think about the world. And let's go from there. Please feel free to take pictures of anything, share anything you like. And we're going to start like that. I'm going to start the talk talking about my group. And again, we're going to talk about philosophy for a little bit. I think I've made it a very intentional choice over the last few years to do a better job after I got some grants and made sure I was going to have a job, to make sure our lab was really one that represented the world and work to diversify it. And I'm going to put up our statement that's on our lab website. And I think it deserves a moment just for us to think about it together. And I'm going to read the first paragraph out loud just because I want to, and I have the microphone. Our lab's committed to creating and maintaining an environment in which all are welcome and respected, one that's inclusive of race, gender, faith, sexual orientation, ability, and socioeconomic status. We acknowledge many identities are underrepresented in STEM and in academia, and that systemic racism and other discriminations create and have created obstacles for students and postdocs. We strongly condemn behaviors and institutions that perpetuate these inequities, and we as a lab, and I'd say academic community, must act to dismantle the structural and systemic barriers to higher education. You can see there our mission statement. I think it's just worth reaffirming my own and our lab's own values that I think are super important. And I think this isn't, obviously this was over the summer in Cleveland because it looks a lot colder right now, but I think our group is moving and growing together. And really, I'm proud to be part of a group that is welcoming to everyone. And I think if you ever came and visited, you'd agree. Like Wafik said, I have a really varied background. I was a nuke engineer. I was an astrophysicist, failed astrophysicist, failed nuke engineer, or then I was a failed physician, and now I'm a failed mathematician. And I think what I am today is someone who's really seeking truth and seeking to better our world. And I happen to do that through the lens of human health. And so I am going to talk a lot about different methods. Yesterday, you would have heard me talk about stochastic processes on high-dimensional surfaces. I would talk about non-continuum models and game theory. Stochastic processes on regular dimensional surfaces. But today we're going to talk more about systems biology. We're going to talk about some tumor genomics. We're going to talk about some network biology, which I think is interesting to some in the room. And our lab studies a huge range of stuff. It's probably because my parents refused to treat me for my ADD when I was a child. But at the end of the day, it makes me happy to study a lot of things. And I also think the diversity of our thought in the lab is also a strength because all of our weird projects play off each other. And sometimes the greatest discoveries come by accident, right? It's actually pretty rare that I sit down and say, I'm going to discover something today. And I think that's probably maybe something that you guys understand too. But we use a whole bunch of different tools, and I get excited by all of them. But again, today we're going to talk mostly about tumor genomics and some mathematical modeling. Okay, so let's get uncomfortable. This is a literal figure. This is figure 22.1 from the standard radiation oncology textbook called Hall. It's written by, it's an Oxford University Press book, I believe. But this is on page 350 or something in chapter 22, describing the original experiments from which we get our fractionation schemes. Let me point out a few things about this picture that might make you uncomfortable, or at least give you some clues as to why it's in the book. One, the experiments were done in France. You can tell this because the ram is wearing a beret. There's also an Eiffel Tower in the background. Why those are important for this figure, I don't know. Back then, this is the 1920s, we figured out fractionation schemes. The radiobiology of the day was done by fractionated radiation to ram scrotums. And at one dose, the ram became azoospermic. I wouldn't want to be the person doing those measurements. And at a different dose, the ram's scrotum became red. The skin became red. And those two dose levels have maintained and are our standard reference dose levels, something like 45 and something like 60 gray, and then 20 for azoospermia and hormone deproduction. That should make you uncomfortable that we're still using those doses. It makes me uncomfortable. And when my patients ask why I'm going to give them 50 or 60 gray, I get uncomfortable because this is the answer. Yes, we've done clinical trials to show that those work, but there's not really much other basis to why more or less might work for an individual. I'd like to also, okay, there's also a scientific disclosure, or sorry, financial disclosure that's real. A lot of the things I'm going to discuss today, I invented and have a vested interest in. So do consider that as a bias that's real. I try not to be biased, but I'm human. And further, a lot of the dosing regimens and drugs we use in clinic today, we talk a lot about targeted therapies and immunotherapies and CAR T and all this beautiful stuff that's new, but most patients still get good old gemcitabine, cisplatin, carboplatin, drugs that have been around for how long, Rafiq? 50, 60, 70 years? Long time, long time. So I think we should be a little uncomfortable as a group that we're still using those and we're still stuck on those. Okay, here comes a little bit of philosophy for just a few minutes so you can think. I really think about cancer and infections as complex adaptive systems. I'm a physicist, I like complex things, but really these maladies, if you will, change and evolve within us on multiple timescales through a series of different ecological and evolutionary mechanisms and forces. And so I like to think about, and I'm going to go through these timescales one at a time and just flip up some ideas that we play with to give you a flavor of some of the things we do in lab in case you want to talk about them after the talk. So what I call the slowest timescale or the evolutionary timescale is represented on the left. And the way to read this little picture is the evolutionary biologists will recognize the left-hand part as a Mueller plot. So this triangle over here is something called the Mueller plot. The way you think about this is when each color is a species. So most evolutionary biologists think like, a finch with a long beak, and then from that comes a finch with a short beak to eat a different kind of seed, for example. You can think of this like clonal dynamics in cancer. Maybe you've seen pictures like this in tumor evolution papers. And then the height of it, the thickness of the ice cream cone is sort of the tumor size, if you will, the population. So what you have is a single clone that starts here of one genetic identity and that grows in a mass that begins to have clonal evolution, which I might call speciation events, right? You have clones of cancer cells that do different things. And over time, you have a heterogeneity that develops at the time of when we biopsy a patient at max diversity. This slowest time scale of mutation, which I call the evolutionary time scale, we think a lot about, and I talked about it at length yesterday and won't today. But the ideas we play with in mathematics are the ideas of fitness landscapes and seascapes, some of which is really actually were born here in the evolution departments up on the hill. See my brown lingo up on the hill? I just learned that yesterday. Brown. And then we also play with, this is Ishan King, who's an MD PhD who just graduated, sorry, he's an M3 now, who really has been using these same mathematics, but thinking about how to mix that with PK/PD models. So how to bring them toward medicine, away from the evolutionary biology labs, not that they shouldn't be there, but toward the ideas of clinical ideas. So he has a paper coming out very soon in Science Advances where we're trying to bring this idea to clinical dosing regimens. I'll send it around when it comes out. Should be just in a few weeks. You can see the preprint here. And then we also used some ideas from quantum information theory, and a paper we published a few years ago in Nature Physics, using ideas that people have been using to sort of push around ground states within quantum systems to try to move from one state of a tumor to another, from a sensitive to a resistant state. Strike that, reverse it. From a resistant to a sensitive state. You wouldn't want to push it the other way. We do that enough. Then there's this next time scale, which I'm going to call a clinical time scale or an ecological time scale, where we already have set up a diversity. A tumor that you see, maybe like the ones we see, where you biopsy it and there's 10 or 15 or 100 different types of cancer cells within that. And on this scale, we really think about the tumor more as an ecological entity. So think about a forest. You don't need to have mutations in one of the tree species for there to be differential tree population dynamics. You could just have a new parasite come in, right? An emerald ash borer comes to Ohio and we have no more ash trees. It wasn't because the ash tree mutated. Actually, we kind of hope it does mutate to get rid of the emerald ash borer. But in these ecological dynamics, we also think hard and we use tools called evolutionary game theory, which again has been used in theoretical ecology for many years to understand how ecosystems evolve and work together. But we're using that to think about something called adaptive therapy in cancer, which is sort of a new idea where we're treating the individual populations differently and working together with that ecosystem that is the tumor. Here's some papers you can see. This is a recent one I'll highlight maybe later. And then the time scale that we're going to focus on today, I call sometimes the now time scale. What can we do today with the information we have in our hand? I don't want to wait. So my patients don't really, I mean, they do care, but the question they're asking me isn't, what does the ecosystem of my tumor look like, doctor? The question is, are you going to cure me? And how are you going to do that? My leg hurts. Can you help me feel better? What should you do today? And that's what we're going to focus on today. And this will work. A lot of the work is going to be from Jessica Scarborough, who's my first MSTP grad. She's an intern now at UCSF. Christy Lynn is a fifth-year PhD student with me. Andrew Dewan is now a neuro-oncologist at the Cleveland Clinic. He's my very first PhD student in Oxford, and Masahiro. And then I work together with some friends from Moffitt as well, Javier Torres Roca, who just popped up there. And these are some papers that we're going to highlight today. So in the radiation world, there's a series of papers in the Lancet Oncology, and then a newer one in NPJ Precision Oncology that Jessica did. Okay, that's the end of the beginning. Does anyone have any philosophical questions? Please.
Now, there is also another part of philosophy, and you already have existing pool of diverse cancer cells.
Yes.
And with the selection question, the evolutionary question, you select those that so on.
Absolutely.
Yours start with a single cell and longitudinally evolves from the reaction to the same question. So what are your thoughts about that?
So my thoughts are exactly correct. So what this picture doesn't yet include us treating it. This is to up until the moment we begin to perturb the tumor. I completely agree the minute we apply selection pressures. And what I forgot to describe was the right-hand side of the plot. If you guys know what Surkos plots are, right? We link this gene to that gene or this gene to that gene. I'm using the idea of a Surkos plot to do a GP map, a genotype-phenotype map. So in this case, maybe this blue one has a sensitivity to this specific drug or that specific drug. So the minute we start perturbing this ecosystem, you're right, this heterogeneity is going to collapse to something new. Probably a less heterogeneous, more homogeneous resistant population, likely. But you're right, I haven't shown any of that post-treatment dynamic.
And just to follow up on that, is that so this is looking at the tumor cell itself, but you've been talking about tumor ecosystem.
Mm-hmm.
So the ecosystem is not made of tumor cells alone.
Correct.
So how does the modeling accounts for those other players who are not tumor cells, but they're also co-evolving, but not necessarily kind of recreating kind of a mutation and heterogeneity?
You should have come to my talk yesterday. I totally agree. And we do consider that deeply in, especially the ecological modeling. We have cancer-associated fibroblasts that we're interested in, immune cells, other aspects of the tumor microenvironment that are included in those models. We're not going to talk about that today, but I think it was recorded. I'm not sure. I'll send it along. Or you just have to catch me next time at an evolution meeting. Okay, so that's the end of the beginning. Now we're going to go into some nitty-gritties about radiation oncology. bear with me. Rad-onc, first of all, is awesome. If you don't know anything about it, great field. If you're a med student out there, pick it. It's awesome. We are oncologists first, and then radiation and physics folks second. I think it used to be a little bit the opposite, but it's a great field. For the med students, it's a 5 years after med school, 1-year internship +4. That we take clinical physics and radiation biology, radiation physics boards, and clinical boards, and then oral boards a year after. It's a great field. I think we think deeply about all the oncologic disciplines. At Tumor Board, it sold me because rad-oncs know about the surgeries, rad-oncs know about the chemo, and rad-oncs know about the radiation. We really have, because it's almost always a multidisciplinary team. What I do in sarcoma is awesome. I work hand in hand with surgeons. I go to the OR. I have a procedure that I can do in the OR to deliver radiation very locally. I work closely with my med-onc colleagues to do combined chemotherapy, chemo rads, all kinds of stuff. But let's talk about some details about radiation. So I love this slide because I put this slide up once in a talk I was giving in Peter Mac in Melbourne, Australia. And this cool wood carving you see is a really nice representation of how we used to give radiation from the front, from the back, and then sort of starting to sculpt more and more 3D conformal beams from different directions. And now this IMRT on the bottom, which is intensity modulated radiation therapy, is multiple beams. And actually, even today, we do what's called volumetric arc therapy, which is beams from every direction. And you can imagine each different projection of a tumor shape is basically the way the silhouette looks of the beam angle as it goes in. But what's really neat, so you can see that this progression here of that wooden doll. So the cool thing that happened at Peter Mac was the guy who carved that doll was in the audience. And he was like, hey, that's my wooden figurine. It was pretty cool. But so, I think that this is the single most common therapeutic agent in all oncology. No other drug is given, the only other thing that's given as commonly as radiation. What, does anyone know who wasn't at my talk yesterday? It's a knife. Surgery. So we still cure a ton of patients with surgery, early stage patients. And the frustrating thing is that today, the practice of radiation oncology, the dosing strategy is empiric. It's physically personalized, right? Each anatomy is different, but the dose is the same. So I'll spend hours personalizing the shape of the dose for a given man's prostate or a woman's breast cancer or a sarcoma in the thigh, but I don't think at all about the dose. It's the same for everyone. And that's just the original idea of a digital twin. And so I think to me, that's really frustrating because, 20 or so years ago, we sequenced the human genome. Actually, how long has it been now? 25 years ago? 25 years ago, we sequenced the human genome. The cancer genome followed quickly after. Targeted therapy has been running, and precision medicine has been all the excitement, and we've been truly helping patients immensely. And my field's just left behind. But why is that? So let's talk about a couple things about precision medicine. So right now, if you had to guess, what percentage of patients do you think are helped by this notion of precision medicine? So let me ask that a different way. What percentage of cancer patients who are going to receive cancer therapy get their tumors sequenced and then have something done differently than the standard of care based on that? I'm going to do a show of hands. How many do you think 50% or more? How many do you think 30% or more? How many do you think 20% or more? 10% or more? It's probably right around there. The real answer is underneath that blue, because I got yelled at once giving this talk at Sloan Kettering, is about 8% of patients have their tumors biopsied and have something done differently because of that. So at Sloan Kettering, they claim it's more like 40%. Maybe that's true, maybe it's not. I split the difference and called it 25 so I could keep going on this slide without getting yelled at. But in 2018, it was estimated that a quarter or less patients benefited from any genome-driven care. Now, why is that? Well, because you really right now, you need an actionable mutation, right? You need a mutation that a drug affects. EGFR, ALK, ROS, RET. The list is growing, but these are still the minority of patients. But tumors aren't just mutations, right? You can have heterogeneity that's based not just on flips in the base pair. You can have epigenetic changes. You can have gene expression changes that's driven by mirrors. You can have circularized DNA. You can have all kinds of things that make tumors behave differently, that affect that genotype-phenotype map. And at the end of the day, as oncologists, what are we doing? We're applying a selection pressure onto that tumor. And what does selection act on? Everyone go back to 5th grade biology. Does selection act on genotype? No, you say. Selection acts on phenotype, right? I don't care what kind of mutations you have. If you're a bird, I'm going to catch you with a net. Well, I mean, who catches birds with a net, really? Papageno, I guess. But what about radiation therapy? How come none of our patients do? If you ask this question about what percentage of patients in radiation oncology have their care affected by genome, it's zero. Zero. We know that HPV-driven cancers are more sensitive to radiation. We don't do anything about it. We run some trials here and there that have all failed, and I'll talk about that. We know that EGFR-driven cancers behave differently. We don't do anything about it. It's frustrating to me. It makes me uncomfortable. So let's talk about this, right? So what is it that's really important here is thinking, and I'm going to shift everyone's focus from genotype to phenotype during this talk, I hope. So if you think about on the left, we have a cladogram, a cartoon cladogram, if you will, of animal life. And we all know that bats and rodents are animals and more similar to one another than are lizards and birds genetically. But phenotypically, if the phenotype of interest is flight, the genetic distance is huge, but the phenotypic distance is right on. I'm going to argue that similarly, we shouldn't be thinking about genetic distances between cancer cells. We should be thinking about the phenotype itself. And so in this case, what we're going to do is try to focus in on those phenotypic similarities, even in the presence of genotypic dissimilarities. Because what I do is select, and I select on phenotype. But if I'm blind to it, I won't be able to. Okay, that said, there's a bunch of signatures out there that aren't genomic based. So Oncotype, MammaPrint, Decipher, these are all expression arrays, right? And these are in practice. Almost all women get Oncotype now for breast cancer. MammaPrint if you're in Europe or elsewhere. And then men are starting to get Decipher more and more commonly for prostate cancer. But what do those do? Those focus on a disease, breast cancer, prostate cancer. Sometimes there's some head and neck ones, I think now. And they focus on the benefit of any adjuvant therapy. They don't say what to do. They just say, be aggressive, be aggressive, right? So no one remembers that from like when you're in high school. Anyways, that was a cheer we had. But the idea here is that they don't tell you what to do. They just say you're at higher risk. So they're really prognostic transcriptomic scores. But what I'd like to do is switch that script. So instead of talking about, I'm going to put this up. This is a talk in two acts with one theme. So phenotype's the thing that will reveal the weakness of the king. Does anyone recognize that quote, who I stole it from? The play is the thing that will reveal the weakness, madness of the king, is it? I can't remember. Anyways, it's Hamlet. My dad's an English teacher. Had to sneak it in there. But we're going to try to move away in Act 1 in radiation and Act 2 in chemotherapy from that to the new focus, which is focusing on a therapy, so radiation in the first part, and its benefit for any person with any disease. So I'm going to try to move away from this idea that lung cancer and head and neck cancer are fundamentally different and move toward an idea that the genome of any cancer should be, or the transcriptome in this case, should be able to tell us about its response to a therapy that I'm interested in. And again, we're going to start with radiation therapy. So what do we do right now? We lay patients on the table. We spend a lot of time thinking about the anatomy. We have medical physicists who are brilliant professionals who work with us, who calibrate these machines to within 1% every day. These machines deliver exactly what you say they do, precisely the same. The problem is that's not what matters. The problem is the effect of that dose. And here's that, literally, not hyperbolically, these are my first 3 patients as an attending. They all had sarcomas in their right thigh. One was African-American, one was Irish, and one was of Eastern European descent. I gave them all the same dose. They had massively different normal tissue effects. Anyone who's practiced radiation oncology for more than a day knows this is truth. No one expects patients to behave the same, and yet our null hypothesis is still that they do, which is, again, frustrating and dissonant. I know everyone behaves differently, normal tissue and tumor, yet I treat them all the same. That's frustrating. So I'm going to argue that we should be thinking about how to measure the biological effect of our radiation. Obviously, we still need to be precise about the radiation itself. Okay, fun story. My second week of residency in 2009, this paper came out. I was a PGY2 at Moffitt Cancer Center, where this paper came from. I remember very specifically, my wife and I had just moved to Tampa, and the Red Journal, which is our scandal rag for radiation oncology, showed up. And that's when we still read journals because there were no computers in 2009. That's not true, because I still read journals because I'm old. Back to back, this paper and another one came out, and I thought, holy moly, how lucky am I? First week of residency, and I'm witnessing the dawn of personalized medicine in my field, and I'm present in the place where it's being born, and I'm going to learn to do this in a new era. What year is it? 2025, and we still don't do it. And this paper's been around for that long, the basis of everything I'm going to tell you, because we're comfortable doing what we're doing today. So what?
Are we comfortable in doing what we're doing today, or people are not, like, for example, as you rightly said in the beginning of the presentation, that is something new. Now, both, not only the person who's delivering, but also the person who's receiving. Both should be ready.
Both need to be ready. That's true. And I'm not arguing that we jump willy-nilly into new things. Of course, it's medicine, it's people's lives at stake. But I do think that we need to think about starting to at least measure the right things.
Most importantly, I've been thinking, like, one of the things is education, public, to be aware about the benefit of this.
Yes, agreed. And I think it would be irresponsible of me to advertise it at this point because it's not even yet available. But I do think that when our trials show this works, or if I should say, I hypothesize that this works, I'm hoping our hypothesis is validated, then I think that we will start telling the lay public about it, right? I mean, I think people do a good job of this. Come get your tumor sequenced in our center because we'll act on it versus another center that won't. So I do think this would be a draw in particular. And I do think people are interested in these trials.
So how much of this is just reluctance of the old guard?
I think a lot. I've had a lot of, so Javier there has spun a company up around this that I put on our disclosure slide. And it's amazing when the potential investors call KOLs, who are usually old guard, they say, oh no, we don't need this. Whereas you talk to residents who've been reading this since they trained, why the heck don't we have this? When I present this at Estro and Astro, everyone says, why can't we do this? So there is a activation energy that's hard to get past. I think it was Max Planck who was famously quoted by saying that science advances one funeral at a time. Hopefully there's not any more patients, but instead old guard standing in my way, in our way, in the way of progress. I won't mention any names. No one in this room would stand in my way. Anyway, so what Javier came up with is this is back when the NCI 60 was all the rage. You could get it was actually 50 something solid tumor cell lines. You had their Affymetrix arrays available publicly. Thank you, NCI. And then what Javier did was a very, very simple experiment, which is a common in radiation oncology, is a colony forming assay. You plate a bunch of cells, you radiate them, and you see how many survived. So there's a surviving fraction that you can measure. And in radiation oncology, we usually measure that at a number of different doses. So on the x-axis, you can see, this doesn't work. On the x-axis, you can see dose, and on the y-axis, it's semi-log. You can see the remaining surviving. And SF2, the surviving fraction at 2 gray, is a super important number because our standard fractionation is 2. So keep that in mind. We're going to play with that a lot. Oh, thanks. We're going to play with that a lot in the next thing. So what he did was he measured all 60 of these cell lines. Oh, does it work now? Oh.
No, it's working.
Okay, thanks.
You need to push it hard.
Okay, I will. What we needed to do was, oh, there it is. It's just light. It's okay. I'll just dance it out. This idea of surviving fraction at 2 gray is really where we moved. Oh, there we go. Thank you, Mani. What we're going to do is develop a genomic classifier to teach us oh, wow. Oh, there we go. Thanks. Or I could do like, my son's super into recreating the laser battles in Star Wars. There we go. So what we're going to do is we're going to develop a genomic classifier to tell us at standard fractionation, 2 gray per day, how much is left over. So we started with, because this is also 2009, everyone was into these hairball diagrams. How nice is it not to see these in papers anymore? Yes, it's nice. Exactly. So this is the top several hundred genes by linear regression that were based on the outcome of SF2 in his cell lines. And then most importantly, back in the day, if you guys remember this time, so nowadays, if you put like the word nano or immuno in your grant title, you get like 5 bonus points because it's a sexy topic. Back then it was networks. So if you did network biology, it was plus 5 points. And so what he looked at back then was the degree of these nodes. And I still think this makes sense. So the degree of a node is how many other things does it talk to? And so the idea is if you have a gene like this one with lots of ins and outs, it must be talking to a lot of different things. It's an important gene in some way. So what Javier did, which I think is always smart, is he picked the top 10 based on degree, and he collapsed the signature down to 10. Here they are. Don't worry too much about what they are, but I think the idea is that this is really an evolutionary problem, right? All of us evolved, well, populations evolve, people don't, individuals don't. But our species evolved from a single common ancestor. That all has been bathed in low doses of radiation for the entirety of geochemical time. So radiation response, it's not crazy to think, is conserved across many species and many tissue types. And the idea here is that these genes serve as sort of canaries in the larger coal mine of that entire signaling network. There's lots of different ways you can mutate a signaling network to get more or less radiation sensitive. These pick up what I think I'm going to hope to show you is a signal that is helpful. Whether or not they're perfect or causative, I don't really care. As long as the signal that I'm developing from them can help me understand what to do, I do care. Okay, so what Javier did, oh, please.
You know, some of my favorite genes are not in those networks.
Yes, exactly.
P53.
Yes.
So what's up with that?
So common great question. Where the heck is my gene? Right? Where's NRF2? Where's KEAP1? Genes that have been,
ATM,
where's ATM? Where's 53? Right? Sorry, where are Rafiq's genes? So, but I think that isn't the point here, right? Those are important, but those talk to these. So if you have perturbations in your gene of interest, it's going to perturb one of these and perturb my signal. Same being, that's why the out degree and in degree of these is such an important thing, because they talk to almost the entire network. So perturbations anywhere in this network, your gene's somewhere in there, I promise, just squint, is going to affect downstream in knock-on effects one of these genes that we are signaling. Again, these are like antenna. It's like I keep doing that, sorry. For predicting weather, we don't measure temperature everywhere. We don't measure wind speed everywhere. We measure it a lot of places, but we interpolate in between, and we think we understand by equations of motion how they relate to one another. Similarly here, we think that these genes are picking up a signal, and then we're able to just use some mathematical analysis to come down to a single signature, lock it in place as of 2009. We've never trained it or changed it since, so everything you see from now on is stuck, which is good and bad. And what he did to begin with, before I got involved in 2014, this is 2012, was he started going through existing prospective trials and asking whether this signature can tell who does well or who does poorly with radiation. So on the left, this is an NSABP trial, the number of which I forget and I don't care about anymore because I'm board certified. You can't ask me that question anymore. But on the left, we see a cohort of patients who received radiation, women who received radiation, and you can see that this signature nicely dichotomizes good and bad outcome. On the right, you see a cohort of women who did not get radiation, and the signature tells you nothing. So suggesting this is a predictive biomarker for this specific intervention of radiation oncology or radiation therapy. This was then done in many cohorts that have shown that it's validated across almost all diseases. There's a couple p-values in there that don't quite make the cut, but they're small studies. But I think the problem up to this point was that this is an indicator, yes or no, of radiation good response. Dichotomized at some sort of random cut point. You know, it's a little bit of p-hacking, but still there's signal there. But the problem really is like, okay, let's say you get glioblastoma and it's resected. We know for sure that radiation improves survival. So now if I biopsy your tumor and say you're a bad responder, what do you want me to do? Not give you radiation? No one wants this information to this point. It's not really helpful because we're not going to withhold treatment that we know in large trials has been helpful, but we also know something that we didn't know before. So our aha moment came in actually 2014. You guys know the few weeks before your deadline for your big meeting, and you're like, oh gosh, abstract deadline. What do? You like run around to all your students and be like, hey, got any ideas? Want to write an abstract? So I'm sitting in my resident room just trying to do my work, and Javier comes running around saying, hey, you just got back from a PhD. We got an abstract deadline in a couple of weeks. Want to help? And so we started looking at this data. So this is data from a large tissue repository at Moffitt Cancer Center. It's about 10,000 patients. And if you measure across the diseases, and you look at those black dots, that's the median of each of these distributions. And it goes from radiation resistant up here, sarcomas, gliomas, to radiation sensitive down here, curable, head and neck, but it's not perfect. But the medians kind of line up. But look at this massive heterogeneity within each population. Right now, what you're looking at is patients with liver cancer who are more resistant than some patients up here with a sarcoma. Now, obviously, that doesn't make sense if you're in the disease-focused silo, but what we're seeing is that there's a heterogeneity here that we're not taking advantage of. What we've done before is just dichotomize, and what we're looking at is a continuous variable that we were throwing away. And so this is all summarized in our 2017 paper. Please. Great question. So these data in particular are surgically resected primaries in this tissue bank. I will show you data from other things in a little while, but these are surgical primaries of patients who are ostensibly cured with surgery. We have done, I'm not going to present today, but we've done papers on metastatic disease and differential sensitivities in those between colon mets, liver mets, and lung mets in colon cancer, among other things. But these are primaries. Okay, so let's go back to this. Remember, we have this simple SF2 formula. I'm going to show you really quickly. This is a canonical equation in radiation biology, and it says that the surviving fraction is this exponential formula. And this is just in that same textbook with the French RAM. Don't judge. It's an important equation we use all the time. And these two parameters, N is the number of fractions that you give, so how many times you treat a patient. D is the dose for each fraction. So these are perturbable by a physician, right? I can change how many times I treat you. The alpha and the beta are intrinsic cellular parameters. They're called the linear and the quadratic term because they are linear and quadratic, but they're presumed to be biological intrinsic parameters of tumors. Most of the time, what we do is we assume for a given disease, a specific alpha and beta. Lung cancer, 3.4 or whatever. But what I'm showing here is if you kind of rearrange this equation a little bit, remember what Javier measured was the surviving fraction. That's what that experiment to begin with. And what that equation here lines up with is a prediction for SF2. So now it's SF2, so N is 1, D is 2. I gave 2 gray once. And now I know the surviving fraction, 0.7, for example. And if I rearrange this equation, I can now solve for a patient-specific, tumor-specific alpha. At low doses per fraction, which 2 is, alpha is hugely dominant in this equation because alphas are on the order of 3 or 5, and betas are much lower, on the order of way below 1. So what we're doing is assuming that beta kind of doesn't really matter that much. So to leading order, we now have a personalized predictor of radiation response. If I can measure this, so I can measure this RSI with a tumor biopsy, I can plug it into this equation, and I now know what a patient's alpha is. So this is now something I can act on. The first thing that's scary about that is now I know that giving 2 gray doesn't do the same thing for each patient, because depending on what your alpha is, now I'm going to show you the way to calculate GARD is just a simple equation with alpha in it. So if I give 2 gray, for example, depending on what your RSI is, that number I get from the tumor genomics, you can have massively different effects of the same dose of radiation therapy. So 2 gray, which is our standard daily dose, can be orders of magnitude different from patient to patient. Remember those ladies' thighs I showed at the beginning? There we go. One of those ladies I might have overtreated the heck out of, and the other one I might have undertreated, even though I gave them the same, right? And so what we're pushing for now is a change in the paradigm of our dosing, the way we think about it. So instead of giving 2 gray per day times 30, for example, which is what we do all the time, let's instead prescribe to an effect. I need to get this much biological effect to cure a patient. That might be a very different dose from patient to patient to patient based on what their intrinsic sensitivities are. So moving that thought paradigm from what we give to what the effect of it is what we're trying to push for. Please.
And you're saying the beta
I'm saying the beta is a problem for tomorrow because right now we have none of them. But you're right. So estimating a 2-parameter equation, it gets a little bit more difficult in cell lines. We are working on that, but I think that the way forward for that might be a different model entirely. I'm not sure that the the beautiful thing, there's a conversation I've been having the last couple of days. There's the tension between simple and realistic, right? And I think for now, the good news is that all the statistics I'm going to show you in the next few minutes of survival statistics are all like log rank and things like that are all preserved because the rank of everything, no matter what you put in for beta, the rank will stay the same between one patient and the next. If you have, you see what I mean? So it ends up being monotone and the stats work out. But for now, we're kind of, we call beta, we just assume a small number and leave it constant for everybody.
But the RSI, you said that the genes in there hasn't really changed to 09. Is that to keep the model simple, or there's just nothing that came out that needs to be added to the model?
Great question. So she asked, why haven't we changed the model, the genomic model since 09? Two reasons. One, if we start changing it paper to paper to paper, no one's going to trust us. That's the most important. The field, after 15 years of presenting this at major meetings and publishing in Lancet, is finally starting to believe that this model has signal. If I start tweaking it every time, they're going to be like, now you're really p-hacking. And I probably would be. That said, my goal is to implement step 1, which I'm showing you today, which I think there's enough signal to implement today and help people, and immediately start trying to learn a better signature. Probably disease by disease, because they probably are slightly different based on intrinsic signaling within the disease. But right now, I think if we pull this out, it's going to ruin trust. Money.
Along the same line, is there anyone, for example, that's something similar where you do the pre-biopsy and treat and collect post-biopsy and ask the question, like, what did I start with and what I'm ending up?
Great, yeah, great.
Based on that, maybe you can refine that RSI.
Great question. Mani asked about pre and post, and we're doing that right now under the U54 that Wafik talked about. So there's a Robin, Radiation Oncology Biology Integration Network that the NCI put forward, and our center has one of these U54s. And my project within that, project 2, is specifically looking at the change of this during radiation. What's at the end, what's at the beginning? But the answer, everything I'm going to talk about today is pre-treatment, which is also actionable in the time frame of radiation, right? Because what we do is we scan in a future world, we scan biopsy, plan dose, deliver dose, but you can change in the middle, but it's tough because the time scales are hard.
For RSI, is it consistent within a patient, or is there heterogeneity within the tumor itself?
Great question. It's probably heterogeneous within each tumor. It is. We've done it. So it's surprisingly not wildly heterogeneous. So there's a trial coming out pretty soon, a prospective trial we ran in sarcoma, which is a massively heterogeneous tumor, even on imaging, right? It's like T1 bright over here, T2 dark over here. It's crazy. And what we did was we actually associated imaging-based habitats to genomic changes and dosed spatially the tumor differently, and it worked. That said, most of these data are single biopsies, and there again is signal. Yes, for sure, we should do spatial stuff. You know, I'm an imaging guy. I would love to eventually have a radiomic map that just tells me the sensitivity. We don't have that right now, but that's a future direction for sure.
The same question for metastasized people.
Yeah, great question. Are all METs the same RSI as the primary? Probably not. And as we move toward oligometastatic treatment being more and more common, that's a huge future direction.
Can you comment on the actual delivered dose, depending on depth, and if it's behind bone or going through air?
That's what we're amazingly good at, is making sure that's correct. So the delivered dose, if there's bone in between it and the beam, we give more from the side where the bone's not involved. If there's air gaps, we put it in different directions. That's why we treat from sort of every angle. So the delivered dose, we've got that problem nailed.
So part of the reason I ask is we also radiate mice.
We do.
And we don't do those things.
We do not. We do not. There do exist small animal radiation platforms where you can, but most of the research, we should talk about that. I have deep feelings about, okay, I need another disclosure slide. I also recently invented an animal irradiator for a different company that I'm not talking about today, but I'd love to share a brochure with you. It's got a better planning system than the existing ones. It's simple. It's beautiful. You guys want me to talk about that? I've got slides about that. Yes, agreed. Animal research with radiation is actually deeply worrisome to me because the dosimetric studies that have been done by independent calibration labs show that hospital to hospital, their irradiators do not do what they say they do. It's actually a little scary. I'll show you some data. Okay. We're going to only do chapter 1 and not chapter 2 because I want to finish here. And you guys have been asking awesome questions, and that's great. But I am going to finish out. First of all, raise your hand if you've heard of the Catan nomogram in prostate cancer. Okay, well, this is my friend Mike. He died last year. He has an H-index of 160. He's one of the most impactful quantitative health scientists ever. Invented nomograms, sat on every NCCN panel, biostats. This is the guy. He also had, he was an economist as a PhD student, and he got lymphoma, a big bad lymphoma in his 20s, in the 80s. And we gave him a mantle field of radiation, which is an old way of radiating all the lymph nodes in the cervical. What do you call the ones under here again? Axillary. I've been in evolutionary biology all week. Axillary, pericardial, and all the ones in the central region as well, in the hilum. And we cured people, but we also destroyed heart valves, lungs. And this is my third friend now to die from, he died from a bad valve that got vegetated, but heart failure just last year. But he's a brilliant statistician and one of my best friends. And he helped me figure this stuff out. So one of the problems we've had over the years, I remember I showed you all those individual studies at the beginning. The problem is it's really hard statistically to put diseases together, to do analyses with things like breast cancer and glioblastoma, because the survival curves are so different. But what Mike helped me figure out was how to do a stratified analysis and pool it so we can basically put everything onto the same, basically normalized hazard functions. So we can get rid of the differences in expected disease length. And what helped, the problem here is that the data we need are tumor genomics, radiation dose information, and outcome. Now you should say, oh yeah, tons of cancer patients have their tumors sequenced. Not patients who get radiation. Why? Because there's nothing to do with it, right? So there's very few cohorts of public data. These are actually all of them as of 5 years ago, where we could get the data. And what we showed in this paper, this is the second paper in Lancet a couple of years ago, is two things. One, if you look on the bottom, this is the dose everyone got. This is a prospectively collected trial. So every single head and neck patient got 70. That makes sense. All the melanoma patients got the same dose except a couple. And there's some spread in each of these others. Breast goes from 40 to 75. That makes sense. But these are the doses. But then if you look up and you run these patients with their genomics through this algorithm, we now see massive spread. So you can't do anything with this, right? You can't say what's the effective changing dose in this cohort because they all got the same dose. But in that same head and neck cohort, we actually now have a huge spread of a predicted effect. So the stats you're going to see in the next few slides are going to compare what we can learn from these differences. So remember, these are real patients, real genomics, real tumors, real outcomes. And what we found was that GARD is associated with survival and recurrence. Now look, all these is a forest plot, so this is hazard ratio 1. Everything on the left means you did better, depending on what kind of outcome you want, recurrence or survival, and then depending on the disease. Now notice everything shifts left, so all of the increased GARD is a better outcome. Many of them are not significant on their own, but pooled they are. Now, one of the editorials written about this article complained that the hazard ratio was only 0.96, which isn't much, but that's per unit increase in GARD. So if I told you for every single extra milliliter of cisplatin, you got an extra 0.96, would you give some more? Yes. And so for 10 extra gray, also we're talking big numbers. The dosing differences were all over the place and not significant in any way. And for patients who didn't receive radiation in these cohorts, there was no change. So the sort of negative control is 0. And so what we see is this a very strong signal here that the genomic radiation dose, the predicted effect, does associate with outcome, whereas the others do not. So the way the stats people want to see this is like this. So you see that for recurrence, you have this strong and significant difference for patients with radiation, without nothing, and the same for survival. And these are some students. Patrick Ellsworth is a neurologist at MGH, and Jeff Sedor is a radiation oncologist at Columbia. They both help with this work when they were med students in my lab. And so here's how I suggest we start using this today. So here's an example of a nomogram that Mike helped me make, and a waterfall plot of sort of what your dose is and what we think the hazard ratio would be. So let's take a patient who comes to your office. Let's pick a disease. Let's say melanoma. And we're going to be able to, let's say standard dosing is 18. It translates for this patient into 18 GARD. Whatever dose it was, whatever their genomics are, it translates to 18. Now you can go look down and say, okay, 3-year progression-free survival, 23%. Now, let's say I increase the dose. Now, all of a sudden I have a 40% survival. That'd be great. Of course I should do that. But remember, increasing that dose might cost a lot for this patient. Some patients, it'll be easy. Some patients, an extra 12 gray will only be an extra 12 points of guard will only be a few extra doses of radiation. Some patients, that might be an incredible extra amount that's not deliverable and not safe, right? So we can start thinking about rationally and powering trials for dose escalation in this way, rather than doing it willy-nilly. Okay, so you know what I'm going to do, because it's top of the hour, I am going to try to figure out how to get out of this mode.
So let me ask you while you're doing that.
Yeah.
So this is based on genes, right?
Yes.
But then there are so many other parameters out there which would kind of probably help predict response, like metabolomics, or if you started with the epigenome, circulating biomarkers. I don't know much about it. I'm just curious, do any other parameters used to kind of dose patients besides genomics?
Your question is bang on. Yes, of course. Simply running an Affy array shouldn't be the beginning and end of our move to personalized radiation oncology. But I think, like the question the lady asked, we should stick with this until it's shown to work or not, and then start layering all the other information. There's a guy at Mass General, Henning, who yells at me every time I give this talk. What about bigger tumors? Don't they deserve more dose than smaller tumors? Yeah, probably, because a surviving fraction of a tumor 10x the size of the next one is more cells, right? And so I think all of these things, clinical factors, circulating biomarkers, other versions of the transcript, DNA, all these things should be considered. But for now, we're still sticking with this until we see it over the line. And I'm out of time, but I just wanted to show one more slide for giggles as a thank you. The part of the talk that you're not going to get to see is this paper here. And I think this is fun because there's a picture of this tweet from Wafiq. And if you look at the date, it was 3 years ago last month. And they were reading with Jacob, when he and Wafiq first met at a journal club in the med school, a preprint of the paper that I was going to present in part 2 of this talk. And what I like about this is a few things. One, again, I'm here and it feels great to be here. Thank you for the invitation, and then for hosting all of you and for listening. But also, it just makes me incredibly proud to see Jessica, my first student, really impacting the world, and also to see the power of the preprint, right? And the comments I got from you helped us and helped our paper get better. And I appreciate that. And looking at the version that we published compared to the version in the preprint it's different, right? So I think open science is amazing. I have stopped using Twitter as much as I used to for personal reasons, but I do think it's a powerful opportunity for us to communicate on a global scale together. This is before I'd ever met Wafiq. 3 years before. No, 2 years before. Yes, sir.
Before you finish your scientific part. One thing you said, is slight increase in the emission dose,
Yeah.
in terms of outcome.
Mhm.
That question brings me to this old movie that I watched when I was a kid, Fantastic Voyage.
Yes.
Since you were a nuclear engineer, shaping people too. Regardless of that story, the nanoparticle negated delivery of radioisotopes or heavy metal. That strategy, how much it helped to increase the focal dose of radiation translating into the.
So first of all, does everyone know about the TV show he's talking about? It was when they injected people into like the bloodstream of a human and they got to like drive around in a tiny submarine. It was a weird show. So bringing that up in a scientific talk is awesome because I was a submariner and I watched that show. But I think are you asking about theranostics broadly and radioisotopes broadly? Yeah, so there is a move now, especially in this world. So one of the greatest, like whoever the heck figured out how to synthesize antibodies, that man or woman deserves a Nobel Prize. Maybe they already got one, I don't know. But antibody drug conjugates and antibody chelated radioisotopes are changing the way we practice oncology, for sure. If you have a tumor with a meaningful epitope and you can hit it with an antibody, like, let's go. Stick a beta emitter on that antibody, stick an alpha emitter on that antibody, stick a nasty cytotoxic like deruxtecan or doxorubicin on that antibody, and you all of a sudden change therapeutic windows massively. You can also affect subclinical metastatic disease. Awesome. I don't think those will replace primary tumor treatments with knives and radiation, because to kill tumors that are 10 to the 12th, 10 to the 11th cells, those theranostics and circulating antibody radioisotopes don't seem yet to be able to get there, but they are absolutely going to be part of our adjuvant armamentarium. Interestingly, again, a totally separate talk that I would have given if this was a medical physics audience would be how to start calculating dose to normal tissues in the setting of personalized medicine, and also in the setting of injectable radiopharmaceuticals that go ostensibly everywhere. We don't even know how to do that. We're behind. So I'm just gonna end the talk there, and I'm gonna just close your eyes. Don't get dizzy. I just wanted to go to my thank you slide. Thanks to all of you for listening. Thanks to the folks that fund us. A lot of the work in our lab started with very humble gifts from families to help us start studying some of these crazier questions before we had enough data for larger grants like the NCI ones, and also the American Cancer Society has been very helpful. But I think it's a real, again, that's a real privilege to be here, and it's a real privilege to share my science and be part of this community, not just here, but globally. So I wanted to say thanks.
So to what extent is GARD, or the ability to predict sensitivity, helpful in proposing or suggesting combinations? So for example, PARP inhibitors.
Great question. So for the Zoom audience, Wafik asked, how is this GARD formalism helpful in understanding combination therapy with chemo and targeted therapies of different kinds? So I'd say a couple different ways. One, there's a paper that I'd love to show you that's under review that looks at the combination of an MDM2 inhibitor with radiation in p53 mutated uterine cancers. And what we're able to do is, if people have all seen synergy plots, we think a lot about if you give drug A and drug B in increasing concentrations and all checkerboard plot, maybe people have heard them, you can calculate synergy scores. We can do similar things with chemoradiation combinations and thinking about how, and part two of the talk was a cisplatin sensitivity signature. And one of the things the last slide I was going to show is, what if we had signatures to more than one therapy? So if I had, this is a hypothesis slide, but if I had a radiation signature on the x-axis and a cisplatin signature on the y-axis, maybe quadrant 2 is sensitive to both. Quadrant 1 sensitive to one and not the other. So helping us quantify those things is the next step. Because I think the problem with existing radiation oncology is it's always yes or no radiation. What I think is beautiful about GARD is it predicts and helps you tune how much. And so we can start asking the question, because wouldn't it be great? So the open question in head and neck cancer is, for whom HPV positive patients can I go down on dose? Because we cure almost everybody with chemo and radiation together, like 96%. But the toxicities are huge, so we want to come down on dose for some. Wouldn't it be great if some patients had massive synergies between their chemo and radiation? It would come down to MSK is actually giving some patients 30 gray. They don't even notice it.
To what extent can you take clinical samples and make correlations? Because GARD came out of clinical data.
Mm-hmm.
Can you take a sample from a patient and make correlations to what you would predict?
Yes, absolutely. So that's the next step. The next step. So the goal is to take a sample, analyze it, and then predict what they need, and then deliver that. So everything we've done so far is retrospective, but there are prospective trials ongoing. But right now we're moving toward that. It's not there yet.
In a perfect world where this is my medicine, where it's a way for every patient to have some type of scientific analysis, what do you foresee that being? There's so many different modalities. Like, what would best inform in terms, maybe just in terms of radiation of this conflict? What would you?
So the question is, what's the future look like for these therapy-directed personalized medicine strategies? I really do sort of hope that we could take some collection of initial data, whether it's transcriptomics, like I suggest here, or a larger collection, like Mani suggested, and really pick the best therapy for somebody. How much and what? It would be great to come in and be like, hey, you just need a little baby dose of gemcitabine and on you go. Or you need CAR-T of this flavor and off you go. Right now, we're largely giving empiric, evidence-based, but empiric, guidelines. Like look it up in the NCCN. It says, give this, off I go. That has nothing to do with the patient in front of me. It's the disease they have and the stage they have. And I would love a day, and we know this, like think about, for the clinicians in the audience, the recent approval of Enhertu for any HER2 positive cancer is really starting to break the mold. Never before has that been done. If you're a drug company and you have a new drug, you go get an indication. This works in ovarian. And if you want to also have it be available for patients with breast cancer, you got to go test it over there. What AZ did, I think they're in HER2, I can't remember. They ran a trial and said anybody with HER2 positive cancer of any kind can try this trial, and they showed it worked in all of them. And I think, if we look at Vivek Subaya's LinkedIn and X feed and Wafik's, more and more we're starting to realize that targets on cells matter more or as much as the kind. So I would love a day when I don't even care where your tumor originated. I just want to know what its genome looks like, for example.
So radiation oncologists love to talk about DNA repair.
Yes.
And it doesn't show up in your network. And I asked you about PARP inhibitors and you just like avoided answering it.
So again, I think the network that we show and we use is more like an antenna for the entire network. Of course, DNA damage repair is important. It's like an entire field. It's also why I can't get RTB to fund this. Sorry, hope no one was listening from the study section. But I do think that's been a problem for us, right? If I put this in front of someone who spent their career studying DNA damage and radiation response, and I show them a signature without their genes in it, I'm not discussed. But I think at the end of the day, we're using this is like a big data kind of method, right? It's almost like a top-down approach. So let's use the two examples really quick. So Oncotype, the way they came up with that list of genes was they got a room full of old guard people who knew a lot, and they all suggested their favorite genes and they put them in the hopper and they made a signature out of that. And it worked. Cool. Because we do know stuff, right? It's true. PARP inhibitor, so the genes you mentioned are truly involved in these processes. And I bet I could have made a signature out of those. The other way to make a signature is an unsupervised way. There's like a supervised way and an unsupervised way. This is an unsupervised way. It was supervised by the concepts behind network biology, not by our understanding, our presupposed understanding of the biology. Those are opposed but synergistic methods or complementary methods of signature creation. We could do the exercise. It actually might be fun to sit down with RTB and ask for everyone's favorite gene and make a signature out of that. But this is the reason Max Dean doesn't like my work, because Keep1's not in there. Moabazi doesn't like it because Nerf2 is not in there. You don't like it because P53 is not in there. I like it. Works. Sir.
Does your network tumor like network correlate with this CTC or some kind of
Are you sorry, is the question?
The question is, if you collect CTC,
Mhm.
so
Mhm.
would
they correlate? Would they correlate? That's our hope. So we have again, the active grant we're doing right now is collecting weekly CTCs. And it was a head and neck grant. We talked about it in our lunch, but for the crowd, we have a grant right now looking at standard of care oropharynx, radiation and chemotherapy. We get pre-treatment biopsy, and then we get weekly blood, saliva as a way to get tumor DNA shed, together with daily CT and monthly PET. The idea being trying to put it all together. Nobody knows right now whether or not the CTCs at the very beginning would correlate with tumor transcriptomics. I hope so, because in the future, I'd love to do just blood or just saliva.
If anybody online has a question.
Oh, they did, but I can't click. Sasha, can you see? In the meantime, go ahead, sir.
in terms of what changing the amount of radiation, if you things that have already been proven by the plasma, using temozolomide and other chemotherapies.
So sorry, did you ask what changing the dose would do relative to?
Yeah, what we have to do in order so that we have to now we put something to sort of let this new system
Yeah.
of different radiation, we give it chemotherapy based on the amount of radiation that's
Correct. So the way we're doing the trials right now, there's the breast trial that's open. We're basically using diseases to start with that have known safe ranges where we know that we could give 60 or 50 in breast cancer, for example, or even up to 75, 74, I believe is the highest they've gone in trials. That range has been shown to be safe, but not efficacious at the population level. Same with lung. We tested 60 versus 74 a few years ago, and 74 was actually detrimental, but we think it's because of toxicities. But the idea is that it's generally a safe dose, that range. We just know that the population level, those if you just escalate everybody, you don't win the day. So we're picking those disease sites to start and then asking if the genomics stratify you to need a higher dose, just give those patients a higher dose. And in our first Lancet paper, we showed a retrospective analysis of a huge boost, no boost breast trial that EORTC ran. And so as an example, that trial took 17 years to read out, 10 years to accrue, 7 years for follow-up. And then 17 years later, we found out that you boost and no boost doesn't matter except for like this tiny sub cohort. If you had stratified it, and it was 17 years and 10,000 women. If you had stratified it genomically, we predicted you could do it with 234 women in 5 years. So the power of not just stratification, but also thinking about it. So the question is, we're starting where we know it's safe to go up and down, and trying there first. And then online, Dr. Bonal says, I might have missed it, but does this dose effect happen in mice as well? Oh, wait. Does your modeling work in mouse strains and seeing specific resistance need higher doses? Additionally, if a tumor were to develop in these mice, would you predict they would need increased dose? So another complaint we've always gotten from study section is we've skipped the animal models entirely. We went from cells to a signature to clinical data. That was because we never got funded to do the animal work, not because we didn't want to. And so at some point needs must, and we move on. And so we did the stuff that was free, which was retrospective analyses. But now that I see the human data working, I don't really want to go back to animal models because they're also wrong in some ways. Maybe that'll be left to the next generation. If step one works, maybe this is how I get beta or how I understand circulating effects or whatnot. But I do think, based on the strength of the papers we have so far and the safe margins we know we have, it's worth trying now in clinical trials rather than going back to animal data.
All right, thank you.